Viral meningitis. So basically, meningitis is the inflammation of the meninges, which is this area in between the brain and the skull. And when it's inflamed, it grows a bit bigger, which means it is pressing on both the skull and the brain, which causes a myriad of issues such as fever, vomiting, headache, tiredness, photophobia, confusion, also potentially more deadly ones such as seizures as well. And so for viral meningitis, this is specifically caused by viral infections. It can include herpes and mumps, but also enteroviruses, which are the most deadly and most severe of the viruses that cause viral meningitis. And so here is a diagram of non-polio enterovirus entry and replication. So first we have the receptors. The enterovirus has all these proteins, and then it will bind to an attachment receptor onto the cell. It will enter, uncoat this capsid, and since it is positive strand RNA, it has to replicate itself into double-strand RNA, which has a negative strand, and so positive strand RNA can then be remade for future packing into capsids as well. Because it is positive strand RNA, it can be directly translated into this protein in the bottom left, which is used to create the replication proteins as well as the capsid. And since many of these capsids are made and many of these new positive strand mRNAs are made, these combine, and so many in this top right, many more enteroviruses are made and released into either the bloodstream or the nervous system, which is where the viral meningitis can take place. So for the viral meningitis, it is caused by inflammation. And so how this happens is once all this viruses has entered the area, it will elicit an immune response. So we have just an example diagram. We have this triangle representing an attachment, an antigen of the enterovirus, which will then bind to a protein on an immune cell. And then we have this oval representing a signal that's going to be transferred into a signal cascade, which is going to produce various transcription factors, for example, which are going to enter the nucleus. And once this happens, these transcription factors or enhancers are again, well, not enhancers, these transcription factors or other proteins that are going to modify the DNA and modify transcription in some way to increase the translation and production overall of these cytokines, like these little star examples. And these cytokines are what's responsible for the inflammation. So we see in this top right area, these cytokines are going to be released into the infected area. And if it is in the if it is for viral meningitis, the meninges are going to get inflamed, which you see this, these lines represent the wall of the meninges. And then once they're inflamed, they're going to become a bit thicker. So it's very hard to flow through them, and it's going to be causing more pressure onto the brain. And so for the most severe possible is enteroviruses. However, generally viral meningitis is not very fatal. And the proportion of viral meningitis has increased, but this is mostly because of increased vaccinations of bacterial meningitis, which is more severe. Overall, the more deadly and severe forms of viral meningitis affect children, which they can have more long-term side effects versus adults, which usually can recover after several weeks. And so for determining that it's not bacterial meningitis, which is the main reason, which is the main way to determine that it's a viral meningitis, a lumbar puncture is used take cerebral spinal fluid and do a variety of tests. These tests are then analyzed into a score-based system using a gram-positive stain, neutrophil counts, as well as various proteins in the CSF to generate a score to determine whether or not it is bacterial meningitis. And if it's not, and these symptoms and still are occurring and other diagnosis has been ruled out, it is determined that it's viral meningitis. As well, for helping for viral meningitis diagnosis, especially with enteroviruses, are PCR tests, which are able to actually confirm based on the DNA or RNA, in this case for enteroviruses, being sequenced, the sequenced genome of these viruses, which then are going to be able to determine for sure whether it is a virus. And since there's a lot of different types of enteroviruses, this can be a bit of a long process overall, because there are many different types of attachment proteins for enteroviruses to enter, which causes all the different kinds of the species of enteroviruses. So, and here are the references.